and Miles will go over some rules, and then I'll introduce Ryan and we'll get started. So my name is Susie Spickle, and I'm the Community Programs Director here at the Harris Center and also one of the naturalists here. And if you're new to the Harris Center, as some of you might be, I just want to say welcome, and we're so glad you're here. If you know the Harris Center really well, I'm also glad you're here. The Harris Center is has been in operation for over 50 years. And through that time, our biggest goal is to help people get connected to the natural world around them. We hope you fall in love with it. And by falling in love with it, you're moved to take care of it. We do that in a bunch of different ways, from helping to protect land with the Harris Center protecting over 24,000 acres. I think we're almost at 25,000 acres now, which is very exciting. We also offer lots of educational opportunities. That's sort of the core that runs through it all, helping people connect to being outside through programs like this, to hikes on the weekend led by volunteers, to programs in the schools, and for people of all ages, from babies in backpacks to people who are in assisted living programs. So a lifespan approach to helping people stay connected to the natural world, which is very exciting. A huge part of what we've been up to lately is also connecting people to community science opportunities, like our salamander crossing brigades or our nighthawk program, or right now we're launching a kestrel monitoring project. You can find out more about the Harris Center and how to get involved by visiting our website. So enough about the Harris Center, and you are all probably here to hear our presenter. Ryan Stevens is a postdoctoral researcher at the University of New Hampshire, where he studies the functional role that small mammals play in the forested ecosystem. He received his undergraduate and master's degree in wildlife at the University of Wisconsin, and I did see that there was somebody who signed in from Wisconsin, at Stevens Point, and his PhD in natural resources and the environment at the University of New Hampshire. I'm really, really excited to hear what Ryan has to say. I kind of got wind of Ryan on New Hampshire Public Radio, where I heard a little snippet of an interview from him, and I thought, I got to get this guy. He's got two things that I love. Well, three things: small mammals, scat, and fungus. Those are three things I'm always interested in, and the health of a forested ecosystem. So Ryan, it's our pleasure tonight to welcome you to the Harris Center and to have a chance to hear what you have to say.
Yeah, so thanks so much for that introduction, and it's really great to join you this evening, and thanks for watching despite it being such a wonderful day outside. So small mammals such as mice, voles, and chipmunks are really abundant consumers in forested systems, and I'm really interested in how their consumption of fungi, especially mycorrhizal fungi, coupled with their habitat selection, so where they live, ultimately influences forest health and ecosystem function. So if you're not familiar with mycorrhizal fungi, they're really important players in forested systems. So there's two main types of mycorrhizal fungi: ectomycorrhizal fungi and arbuscular mycorrhizal fungi. And hereafter, I'll refer to these as ECM and AM fungi, respectively. Now, these fungi are symbionts, or they're important mutualists that help trees grow. So mycorrhizal fungi provide water and nutrients, especially in the form of nitrogen and phosphorus, to the trees. And in exchange, the trees provide carbon to the mycorrhizal fungi in the form of carbohydrates. Now, maintaining diversity of these mycorrhizal fungi is really important. So different fungi might be active under different conditions or different times of the year. For example, some mycorrhizal fungi are really active under drought conditions, while others might be active under wet conditions. And so maintaining a diversity of fungi helps buffer forests from changes or kind of perturbations, which is especially important with climate change. And fungi in general tend to be a little dispersal limited. They occur underground, and they, to help increase their dispersal, they produce spore carps. Now, these might be above ground fruiting bodies, such as mushrooms, which probably many of you are very familiar with, or they might be truffles, and these tend to be below ground fruiting bodies. And I'll talk quite a bit more about those in the upcoming slides here. And small mammals play an important role in dispersing both mushrooms and especially truffles. And so this is also a mutualistic interaction. where the small mammals get this really important food source, which can make up a pretty large part of their diet. And in exchange, the small mammals disperse those spores. And you can imagine that depending on the type, or especially the amount of spores that these small mammals are depositing with their scat, and especially the location that they're dropping those spores, that can have really important implications for tree establishment and growth. Now, this might be particularly important after a timber harvest or other types of forest disturbances where trees are removed from the landscape, and mycorrhizal fungi tend to be greatly reduced once their host plants are gone. And so we need some mechanism for these spores to get from this intact forest back into these managed or disturbed areas. And mammals, especially small mammals, might play a really important role in doing this. And so today I want to walk through a number of studies that I've done that have kind of helped to untangle a bunch of these arrows I've showed you so far. And so the first vignette will be on what factors shape truffle selection. Why are mammals eating these truffles as a food source? The second one will be on which small mammals are the best dispersers of fungi, or what makes a small mammal a good disperser of these fungal spores. I then want to move on to a forest management context and talk about how may small mammal spore dispersal contribute to forest regeneration, or how might we also be able to manage for these mammals to increase spore dispersal. And then finally, I want to move on to carnivores and talk about the role that carnivores may play as secondary dispersers of mycorrhizal fungi. But before I get into these studies, I want to kind of give you an idea where these data come from and how they were collected. And so I did my dissertation work at Bartlett Experimental Forest, which is in the south central part of the White Mountains, a really beautiful area. We have a number of sampling grids that are stratified kind of across the dominant forest types. So we have 4 grids in hardwood forest. These are composed of American beech and red maple. We have 4 sites in softwood forest, which is primarily red spruce and hemlock. And then finally, we have 4 of our sampling grids in mixed forest, which is a combination of all those tree species. And when we started this project, we didn't really have a good sense of truffle diversity. How abundant are they in the region? How common are they? And before I moved out here, I really didn't know a lot about truffles in general. And so when we started this project, we surveyed for truffles across those dominant forest types. And we did this using truffle rakes. We surveyed 768 4-meter square plot, which is about 3,000 square meters, or just over half a football size area that we surveyed over a summer. So this is a pretty big endeavor with myself and a bunch of undergrads. We raked down to a depth of about 10 centimeters or so. And so I think we were all quite surprised when we unearthed over 6,000 truffles of 14 species comprising 7 genera. And it turned out that 5 of those species were actually new to science. They'd never been described before. So I was able to work with a truffle taxonomist and describe several new Elaphomyces species. And this is pretty exciting. Bartlett was established in about 1933 and is one of the oldest experimental forests in the US. And so to find 5 species that researchers had literally been walking on for about a century was pretty exciting, and goes to show really how little we know about what's going on below our feet. We also found really high biomass of truffles, especially in the mixed and softwood forest. So in the hardwood forest, we found about 3 kilograms per hectare. But in the mixed and softwood forest, we found about almost 30 kilograms per hectare. And this is among the highest biomass that's ever been recorded anywhere in the world. So there's not only a lot of truffles out here in New England, there's a lot of biomass available too. We also found that the most common species were associated with eastern hemlock. And as many of you are probably aware, eastern hemlock is a tree species in pretty severe regional decline due to the introduced hemlock woolly adelgid. So loss of eastern hemlock may have some really important implications for loss of truffle diversity and biomass, an important food source for many of the small mammals. So we also trap small mammals on these same grids. We use these Sherman live traps, which are basically box traps. We did this in the summers of 2013 through 15. Each individual was marked with a unique tag, either an ear tag, or if the ear was very thin, we put in a pit tag or passive integrated transponder. So this is the same thing that you would, the vet would put in your dog. It's just injected under the skin, and then we can scan it to tell if we caught it before. From these animals, we also collected hair sample to look at diet. I'll talk about that more in a couple minutes. And we also collected fecal or scat samples to look at the types and the amount of spores that these animals are dispersing. So it turned out that about 98% of all our rodent captures were represented by these 5 species, and they had pretty different habitat associations. So southern redback voles tend to be in softwood forest. Our woodland jumping mice tend to be in hardwood forest or very stemmy forest where it's very thick. Eastern hemlocks, which I see someone had that as one of their favorite small mammals, I would agree with you. They were in mixed forest, they were in softwood forest, they were in hardwood forest, as were our deer mice and white-footed mice. And so these guys are really generalists. They were in all these different forest types. They were also in our bunkhouse. They do really well in a lot of different habitats. And so we were really interested in what these animals were eating. And to get at this, we used hair samples in a stable isotope approach. We used stable carbon and nitrogen. And this is an example of the food sources that are available to the small mammals. And you'll see that they have pretty unique isotopic signatures within this isospace, if you will. So the seeds tend to be relatively high in C13 and low in N15. The 2 types of fungi tend to be relatively high in both C13 and N15, as well as the arthropods. And berries tend to be relatively low in N15 and C13. So we can measure the stable isotopes of the potential food source of these mammals and also of the hair samples, do some adjustments, do some relatively fancy modeling, and come up with estimates of how much fungi these animals are consuming. And what we found was that the southern redback voles, those animals living in the softwood forest where there's a lot of truffles, on average, about 60% or more of their diet is coming from fungi alone. Compared to the mice and chipmunks, which were probably somewhere around 15 to 20% of their diet from fungi. So although the mice and chipmunks aren't eating quite as much fungi as the redback vole, they're still getting a pretty substantial part of their diet from fungi. So we were interested in what types of fungi that they were eating. And so for this, we used microscopy. And in total, we analyzed over 1,200 scat samples over that 3-year period. We freeze dried them, we grind them up, we put them through a filter and spread that spore extract on a slide, and then count the number of spores we see in a number of fields of view. And we were able to idea a lot of these to genus or even species, especially for the truffles, because they have such unique ornamentations and shapes. And so in total, we found 34 different fungal taxa. And on average, individuals carried somewhere between 3 to 5 different taxa. So redback voles and eastern chipmunks on average had around 4 or 5 different taxa. Most of these were truffles. And our mice species had somewhere around 2 to 3. And if we took into account the abundance of these spores, redback voles by far have the highest abundance. All right, so we know that these animals are eating truffles, and we know that there's a lot of truffles on the landscape. But what factors help shape this truffle selection by the small mammals? So as I mentioned, truffles are the below ground fruiting bodies of fungi, especially mycorrhizal fungi. And they've evolved from mushrooms, from having this above ground fruiting habit to fruiting below ground. And this is thought to be a mechanism to protect them from drought or freezing conditions. So if you fruit below ground, you don't have to worry about humidity conditions or what's going on above ground, but you do have to have a different dispersal mechanism. So this transition in fruiting mode has also accompanied a transition in dispersal mechanism from that of wind for mushrooms to that of primarily of mammals and small mammals for truffles. And so truffles have really closely evolved with mammals, and they produce these odor signals to communicate their location and when they're ripe. And they do this in the form of volatile organic compounds, or VOCs. The mammals can hone in on those, that chemical cocktail. They dig up the truffles, eat them, and then disperse their spores in the scat to new areas. And so we were really interested in the factors that influence this mammal selection. So there's a number of factors that could influence this. The depth below the soil surface. Obviously, the deeper a truffle is, the harder it is to potentially reach. Abundance might also play a role. The more abundant a truffle is, probably the easier it is to find. That volatile organic compound, or that VOC emissions that produce odor, are probably really important. Size might also play a role. The bigger a truffle is, the bigger the prize in terms of digging it up and eating it. And then nutritional content might play a role, and especially protein, which is often limiting in the diets of a lot of small mammals. And so for truffles, we looked at Elaphomyces species at Bartlett, and this is a really widespread genus. It occurs on pretty much every continent except Antarctica. It's characterized by what's called a thick peridium, which is this outer layer, kind of like a puffball, and it has this powdery layer of gleba inside, composed of spores. So small mammals dig these up, they eat this outer layer, and they ingest some of these spores along the way. And so we focused on 4 species that are common or present in mixed and softwood forests. And these included Elaphomyces americanus, E. verruculosus, E. macrosporus, and E. bartletti. And so when we looked at the fruiting depth of these truffles, we found that they're kind of stratified across the soil layer. So americanus is relatively close to the soil surface, pretty much right under the leaf litter. Verruculosus fruited about 2 centimeters below the soil surface. And macrosporus and bartletti fruited somewhere between 4 and as deep as 8 centimeters below the ground. And that might not sound like a lot, 5 or 6 centimeters, but that's about the length of a lot of these small mammals. So that would be a long way to dig down, basically their full body length. We also found some pretty big differences in the abundance of these truffles. So verruculosus was extremely abundant. On average, there was about 25,000 truffles per hectare, whereas the other species had somewhere between 700 and about 3,000 truffles per hectare. So based on this, you might expect that the small mammals are maybe selecting for the shallow fruiting species, maybe americanus and verruculosus, or maybe they're going to go after this really abundant truffle, verruculosus, because it's everywhere on the landscape. And so to get at selection, we used the spores in the scats relative to the truffles on the landscape, and used Jacob's selection index. And this ranges from -1, which is really strong avoidance. So for me, this would be lima beans. They're there, there could be a lot of them, but I'm not going to eat them. 0 indicates that you're consuming it relative to its availability. And so for me, this is broccoli. I'll eat if it's there, but I'm not going to seek it out. And then finally, 1 is really strong preference. And so for me, this is jelly beans. I'm going to seek them out and I'm going to eat every last one of them until they're gone. And so what we found was actually pretty interesting. So these are the different small mammals, the shapes here, and the bars here are 95% confidence intervals. And what we found was that americanus, the small mammals, were eating it pretty much relative to its availability or avoiding it, despite it being relatively shallow and easy to access. Verruculosus, that really abundant truffle, relatively shallow. Pretty much all the small mammals were avoiding it, except the redback bull. And I'm going to come back to that here in a couple slides. And then finally, macrosporus and bartletti, those really deep truffles, we see really, really strong selection. So clearly there's something else going on here relative to fruiting depth and abundance. So what about those VOCs, those odor signals? And so this is an NMDS ordination of volatile compounds in truffles. But all you really need to know is that these different shapes here are individual truffles. And the closer they are together, the more similar the volatiles that they have in common. So what you might notice is that americanus and verruculosus have relatively similar volatile cocktails. And bartletti and macrosporus have relatively distinct VOCs that they're releasing. And if I overlay vectors of all the different compounds they're releasing, the length of this arrow is indicative of their abundance. You notice that there's kind of 2 plumes of VOCs that are produced, especially with these 2 truffles. And I could try to read off all the different compounds that they're releasing, but I thought it'd be easier to give you kind of an odor word cloud of what these might smell like, at least to the human nose. So macrosporus has compounds that have a sweet floral fruity aroma, whereas bartletti has a sweet truffle mushroom, fruity, and some floral and vinegar notes as well. And so if I had to describe these actually do smell very strong. Macrosporus has kind of a sweet citrus smell. And Bartlett I smell somewhere between garlic and road tar. It's very strong. And as an anecdote, we had an undergrad cutting these up in a small room, and Bartlett I was so strong that he was actually getting nauseous, to give you a sense of how strong these VOC aromas are. And so these VOCs, the strong odor, despite them being so deep in the ground, likely allows the animals to kind of cue in on them and creates really strong selection. But what about that mass and protein content? So we didn't see any differences in the mass. All the truffles on average are about 1 gram, although there's some variation among or within species. But we did see some differences in digestible protein. So Verruculosis and Macrospores have nearly twice the digestible protein as the other 2 species. And this didn't seem to make too much of a difference for most of the mammal species, except for perhaps the redback vole. As you recall, these guys are fungal specialists, and they seem to be selecting for verruculosis, at least relative to its availability. And so if you're an animal that eats fungus as its main food source, you need to make sure you're getting enough protein content in your diet. So to summarize this section up, what factors shape truffle selection? Well, those really strong VOC emissions likely allow the animals to detect those really deep fruiting truffle species. And although they're deep down, mammals probably don't have to spend a lot of time looking at them, because they can sense those or smell those VOCs probably from a long distance, kind of hone in, dig them up, and not spend a lot of time looking for other species, although they might be more abundant. And then finally, protein content doesn't seem to matter too much for most of the species, except maybe the fungal specialists, those that are eating fungi as their main portion of their diet. So we know that all these animals are eating truffle, but what makes a mammal a good disperser of mycorrhizal fungal spores? So a lot of focus on fungal spore dispersal has come from fungal specialists, and these might be batongs from Australia. They might be northern flying squirrels, especially from the Pacific Northwest, or things like southern redback voles, which I've talked about here today. But there's a lot of other factors other than just how much fungi these animals eat that might influence their dispersal potential. So things like animal abundance and habitat selection might also be really important. So we were really interested in the role of diet, habitat associations, and abundance in influencing spore dispersal. So we know that the small mammals at Bartlett have pretty big differences in their fungal consumption. Redback voles tend to be specialists, where the other species eat fungi on a regular basis, but not to the same extent. We also have differences in their habitat preferences. So southern redback voles tend to be in softwood forests where there's a lot of truffles, where some of these generalists tend to be across all the different forest types. We also have some pretty big differences in population fluctuations year to year. So up at Bartlett, we have masting events where large amounts of American beechnuts are produced in the fall. And in the following spring, we have really big increases in the populations of especially mice and chipmunks. And so many of you might have noticed this a couple of years ago when we had a lot of acorns that fruited in the fall, and the following year there was a apocalypse of gray squirrels on the landscape. So this is something similar that happens at Bartlett as well. And so we took these spore loads and integrated them with abundance and developed some networks. And so this probably sounds pretty complicated, but it's actually not too complicated. We take animal abundance. Here, redback voles are relatively uncommon on the landscape compared to deer mice. We multiply that times the average spore loads in their scat. So here, redback voles have lots of melanogaster spores, whereas deer mice have relatively few. And then we scale that by contribution. So in this case, although redback voles aren't very common on the landscape, they have really high spore loads. So at the population levels, they're doing most of the spore dispersal for this type of fungi. But you can imagine if spore loads are relatively similar, the more abundant species is going to be doing much more spore dispersal at the population level. And so this is an example of a network. On the top here are all the different fungal taxa represented by letters. On the bottom are the mammals, and all these kind of spaghetti strings you see connecting them are the mammal fungal interactions that are taking place. And it's important to note that the length of this bar on the bottom for the mammals is indicative of their relative importance in the network. So here, these two species are doing most of the spore dispersal compared to the others. And so what we found was some pretty big spatial dynamics of spore dispersal across the landscape. So this is the network I just showed you for hardwood forest. And as you'll recall, redback voles, although they're fungal specialists, they're really not very common. And we actually see that some of these other species, these generalists, are actually doing much more of the spore dispersal. If we compare that to a softwood forest where redback voles are quite common, we now see that they're doing most of the spore dispersal. So this probably seems pretty intuitive. You have to be in a habitat to do the spore dispersal, but most studies really haven't addressed kind of abundance and habitat selection when they look at spore dispersal. We also found some pretty big temporal dynamics of spore dispersal among years. So this is mixed wood forest, and this is following a non-masting year. Redback voles are doing most of the spore dispersal, and our chipmunks and our deer mice are doing relatively little spore dispersal. If we compare this to the same forest type following a masting event, we now see that redback voles are not doing much of the spore dispersal, and our generalists are actually doing almost all of the spore dispersal in this network. So we have some pretty big temporal dynamics year to year that are driven by population fluctuations. And during these years following masting events, we see that more fungi are dispersed. And we also see higher diversity in the networks. So in 2013, in the fall, we had the masting event, and these generalists become very abundant in 2014. And we see much higher level of diversity within these networks, indicating that more species are taking place in this spore dispersal dynamic. And this is really important because different small mammal species live in different locations or different microhabitats. And so the more animals that are taking place, the more chances that these fungi are going to be dispersed to new areas. And so to summarize this section up, which small mammals are the best dispersers of mycorrhizal fungal spores? And that it's kind of maybe a trick question. So there's some both spatial and temporal variability in this spore dispersal. So although specialists like southern redback voles are consistently important in their favorite habitat in the softwood forest, the generalists also tend to be really important in areas where these redback voles aren't, and when their abundance is really high. And the timing of this spore dispersal of the generalist may be really important. So we have these masting events in the fall. In the following spring, the populations of the small mammals increase greatly. And this is a time also when these seedlings are beginning to establish. And mycorrhizal colonization of seedlings is really important for their growth and survival. So not only are these small mammals, these generalists, dispersing more spores, that they're also doing at a time that's really critical for the establishment and growth of seedlings. So now I want to move on to kind of a managed forest question and ask how may small mammal spore dispersal contribute to forest regeneration? And so for this project, we moved to Second College Grant, which is way up almost on the Canadian border, about 40 miles away. And this is a really interesting managed forest system. It's part of the adaptive silviculture for climate change, where foresters are looking at ways to manage forests into the future, whether that's different types of cuts or actually bringing in southern species and planting them. And in this area, pretty much all trees associate with either AM or ECM fungi, although different species might associate with these different types of fungi. For example, sugar maple is associated with arbuscular mycorrhizal fungi, and that's a really important tree species economically, both for timber production and also for maple syrup. And ECM-associated trees include things like red spruce and a lot of birches. So it's really important following management that these fungi, both AM and ECM fungi, get back into the cuts following forest management. And so our research approach was first to sample small mammals to look at spore dispersal and the microhabitats that they're living in, where they might be dispersing those fungi to. Second, use the greenhouse experiment to look at how small mammal scat might influence the growth and establishment of seedlings. And then finally using field exclosures that restrict small mammal activity. And so the first part of this project is pretty much done. And the second two parts that I'm going to talk about are still in transition right now. And so second college grant, we took advantage of the adaptive silviculture for climate change patch cuts. And these are about 1 acre in size. And we had these in mixed and hardwood forests. And today I'm just going to talk about sites in the hardwood forest. So at each of these patch cuts, we established trapping webs. So we could sample small mammals from the intact forest all the way into the center of these patch cuts. Again, we used capture, mark, recapture techniques in the summers of 2018 to get the abundance of the animals and collect scat samples. And you'll notice that the players here are pretty similar, although we do have some northern short-tailed shrews, and we also have some meadow voles and some meadow jumping mice, which tend to be more open habitat associates. And so to answer the first part of this question, kind of looking at the fungal diversity, again, we use microscopy. And we found a number of different morphotypes. So these top ones here are all AM fungi, the top row, and the rest of these are primarily all ectomycorrhizal fungal morphotypes. And you can see that they're fairly distinctive. Most of these are truffles. And so in total, we analyzed over 400 scat samples and detected 7 AM morphotypes. And we developed this spore load index that incorporates the abundance of the spores and also the number of taxa that they're carrying. But all you really need to know that the higher this number is, the more types and the more amount of spores these animals are carrying. What we found was that woodland jumping mice have the highest levels of AM spores, followed by meadow voles, short-tailed shrews, and meadow jumping mice. And then finally, deer mice, redback voles, and eastern chipmunks had lower levels. If we compare this to ECM fungi, we find that redback voles and eastern chipmunks by far have the highest spore loads, whereas the other animals at an individual level have relatively low spore loads. And so these letters in the top here indicate statistically differences between the mammal species. But you'll note that although we find differences for AM fungi, really all these mammals are carrying pretty decent levels of AM spores, compared to ECM fungi, where it's really just redback voles and eastern chipmunks that are carrying ECM fungi. So we know that abundance is really important, and we did see some pretty marked population fluctuations, especially of the mice between the 2 years. In 2018, deer mice were really abundant, and in 2019, their populations crashed, and woodland jumping mice actually became quite abundant. So again, we integrated the abundance of the animals with their spore loads and developed networks. And so this is similar to the other networks I showed you, but now they're flipped up vertically. On the right-hand side here, the gray indicates fungal taxa. These are AM fungi. And on the left is the small mammals. And again, the length of this bar is indicative of the relative importance of these animals for spore dispersal. So this is AM fungi for 2018. And as you recall, deer mice were very abundant, and they're doing by far most of the spore dispersal. If we compare this to 2019, deer mice populations crash, woodland jumping mice become really abundant. And we actually see that now woodland jumping mice are doing most of the spore dispersal. So for AM fungi, it really seems that the most abundant small mammals on the landscape are doing most of the spore dispersal. If we compare this to ECM fungi, as you recall, redback voles and eastern chipmunks are really the only ones carrying a lot of ECM spores. We find that redback voles were really the only one in the first year that were entering the cut, and they were doing most of the spore dispersal. And even into the second year, we saw more activity of eastern chipmunks, but still redback voles were doing most of the spore dispersal for ECM fungi. So for ECM fungi, it really seems like, and in this system at least, redback voles are really important. So we were also really interested in ways that we might be able to manage for these forests or following forest harvest to increase spore dispersal. And so for this, we use Microhabitat Associations. And what we found was that deer mice really don't have any microhabitat associations. They're kind of running around the landscape willy-nilly, I guess. Eastern chipmunks tend to be associated with higher stem density. Redback voles also stem density, ECM trees, and importantly, vegetation cover and down woody material. Woodland jumping mice, again, stem density, AM trees. And then finally, our open habitat specialists, not surprisingly, tend to be negatively associated with trees and positively associated with vegetation cover. But what I really want to zoom into is the southern redback vole. That's really important for ECM dispersal. And so how might we be able to manage for this? Well, they're especially associated with down woody material and vegetation cover. And so following a timber harvest, if we can leave some cover, some stem density, and especially the tops of those trees, we can maintain habitat for these species and help them disperse fungi. And so I'll mention too that a lot of seedlings, small seeded seedlings like birches and hemlock also tend to establish under down woody material where they're protected. So not only are we providing habitat for the small mammals, they're also dispersing this to an area where a lot of these seedlings are regenerating. All right, so we know that these mammals are bringing in a lot of fungal diversity. How might this ultimately influence tree establishment and growth? And for this, I set up a greenhouse experiment with red spruce, which is an ECM associated tree. I grew these seedlings with small mammal scat in field soils, and then with small mammal scat that was autoclaved to kill any spores. Then looked at percent root colonization, above and below ground biomass, and fungal community composition. And as I mentioned, this is a bit of an ongoing project, but we have seen some differences where when we add live mammal scat or scat that's not autoclaved, we see that these seedlings grew larger and they had higher colonization than ones that were just grown in field soils alone. I also set up a greenhouse experiment for red maple, which is an AM associate, and again grew seedlings with small mammal scat and then with small mammal scat that was autoclaved. In this study, I harvested pretty recently, but you can see I had pretty good growth of these red maples after only a few months. Apparently, it's pretty easy to grow red maples if a mammal guy can do it. So this is still an ongoing project that I'm working on. And so these greenhouse experiments are great, but they don't necessarily get at what's happening in the natural system. And so for this, we set up field exclosures that were paired with controls. These were constructed of hardware cloth, about 2 meters by 2 meters. They extend from forest reserve areas that had no harvest all the way into the center of the gap cuts. And so this was quite the undertaking. We buried these into the ground to prevent animals from digging them up. And as you can see by the number of rocks, this was a lot of work, but I think it was worth it because we did have a few small mammals try to dig in, but were stopped by the skirt that went underneath. And so these were established in 2018. And in the next couple of years here, we'll be doing some field tests to look at soil fungal communities and spore banks in these exclosures relative to the control plots, and likely doing some seedling growth and colonization experiments, both in a field setting and potentially in the greenhouse. So to summarize this section up, how may small mammal spore dispersal contribute to forest regeneration? And we found that these mammal species are relatively complementary in their spore dispersal. So really the most abundant species are dispersing AM fungi. We're really, redback voles were doing most of the ECM fungal dispersal. So this likely means that AM fungal dispersal is probably stable among years, as long as there's some small mammals on the landscape. Whereas ECM fungi really, at least in this system, seem to rely on redback voles to do a lot of the dispersal. So if we can maintain habitat for these voles, we can help increase spore dispersal and ultimately hopefully help increase colonization and growth of seedlings. And then finally, let me get back to you in a couple of years about how spore dispersal might influence fungal communities and seedling growth. All right, so we know that small mammals are dispersing a lot of fungi and a lot of different types of fungi, but they're doing this at relatively local spatial scale, somewhere between tens to hundreds of meters. But thinking in the context of climate change or larger disturbances, these spores need to move much farther than that. And so I'm really interested in the role that carnivores may play as secondary dispersers by eating small mammals that had eaten mycorrhizal fungi and then dispersing those spores to new areas. And so for this project, I moved back to the Midwest where I'm from, and I saw one person was from Wisconsin here. So this project is taking place in Minnesota and Wisconsin. We've established 3 sites. We have one in Minnesota managed by Michael Joyce at Duluth, and 2 sites in Wisconsin managed by Amanda McGraw of Wisconsin DNR. And so in 2020, despite the pandemic, they were able to get out and trap for small mammals to look at a resource base in terms of the truffles that they're eating, and also a resource base for carnivores in terms of what small mammals are on the landscape. They also conducted road surveys to collect carnivore scats, and were able to collect well over 100 scats of a number of different carnivore species. That we then use genetic techniques to ID these scats to species. And so I'm almost through the small mammal data, and it looks like pretty much all the small mammals are eating mycorrhizal fungi, especially ectomycorrhizal fungi. And hopefully next week I'll be starting on the carnivore scats. So I don't have any results to show you, but we do have some kind of predictions in what we expect in terms of this secondary dispersal. So the 4 main carnivore species that we collected scats were from wolves, coyotes, bobcats, and foxes. And in terms of the distance that these species move, it varies quite a bit. So wolves have relatively large home ranges, anywhere between 400 and 600 kilometers. So we expect that their dispersal capacity in terms of distance is probably quite far. Coyotes and bobcats have home ranges somewhere around 10 to 60 kilometers. So they're probably moving these spores potentially moderate distances. And then finally, foxes have relatively smaller home ranges of several kilometers and likely aren't moving spores as far. However, if we take into account spore loads, which we can think of here as small mammal consumption, wolves in the fall in this region really don't eat a lot of small mammals. We don't expect that they'll be carrying many spores. Coyotes and bobcats eat moderate levels of small mammals in the fall, and we expect that they'll likely have moderate levels of spores. And then finally, we expect that foxes, which eat a lot of small mammals, to carry a lot of spores. And so we did do some preliminary analyses on these, especially of the fox and the coyote and the bobcat. And we did find really high spore loads. Almost well over half of the samples have truffle spores and small mammal remains. So I'm really excited in the next couple of weeks to get into this some more. And so, as you recall, we used road surveys to pick up these scats. So these were scats that were deposited in a road, which probably doesn't have a lot of influence on spore dispersal. And so this past year, Michael was able to bring in scat detection dogs to look for carnivore scats across the landscape. And so we'll be able to get a sense of not only where are these animals depositing the spores, but what spores are in their scat as well. We're also really interested in the viability of these spores. And so we're setting up a greenhouse experiment for this. So these spores now have passed through a small mammal gut and a carnivore gut, and that's some pretty extreme environments. And so we're interested in what percentage of these spores are viable, and do they have an influence on colonization and growth of seeds? So with that, I'd like to acknowledge all my collaborators and supporters, especially my doctoral advisor, Dr. Rebecca Rowe, all the undergrads that helped me in the lab and the field, and of course, my funding sources, especially the New Hampshire Agricultural Experiment Station. And with that, I'd be happy to answer any questions if you have them.
Wow, Ryan. First of all, we will definitely have you back when you get your carnivore result. That was amazing. Really fascinating. Lots of comments in the chat about the interest. And there are a lot of questions. And I think I would like to start with a question that came up more than once, which is, are the underground truffles edible for humans? That was like a really popular question. Can we eat them?
Yeah, so truffles have pretty much evolved to be eaten by mammals for the most part. And so if you're poisonous, that's kind of a bad dispersal mechanism. It probably works once. So yeah, these truffles are edible, but they're probably not choice. I've tried a couple of them, and they're not amazing. You also have to be really careful, because a lot of times mushroom buds before they pop up can look like truffles, and so you really have to know what you're doing before you dig them up. But that said, I have found tuber, which is the same genus as the expensive ones in the Pacific Northwest and parts of Europe, and they're pretty good. The only difference is in Europe, they've evolved to be dispersed by feral hogs. So they get the size of a baseball or maybe a smaller quarter size. But here they have evolved with small mammals, and a lot of them tend to be the size of a pea. So you're not getting the same size that you would in other areas.
So
Yeah.
nobody's going to rush out and get themselves a scat sniffing pig in New England and make a lot of money on pea-sized truffles.
Yeah, probably not.
Yeah.
And actually some of them like Octaviania, to me, it smells like rotting fish. So
Ooh.
small mammals have a different palate to a lot of humans as well. But yeah, so there are some that are edible, but they're probably not the choice ones.
Yeah. I really loved how you use the odor cloud to kind of describe what the flavors were, or the odors would be. That was really interesting. So a question that kind of came up in two places is, do the spores have to travel through a gut system for them to be kind of viable?
Yeah, that's a really excellent question. And there have been a couple studies that have taken spore carps or truffles, fed them to small mammals, and then compared that to just taking the spore carp directly and putting it in the soil. And they got higher germination and higher colonization levels once it passed through the small mammal gut. So there's something either enzymatically or maybe temperature that stimulates these truffle spores to germinate once they pass through the small mammal gut. And there's been a couple studies with mushrooms, and I think one study showed, at least for one of the mushrooms, that germination decreased, and then the other taxa increased. So it's probably kind of specific, but at least for the truffles that have evolved that way, it does seem like passage through the gut actually stimulates them to germinate.
Wow, interesting. So that question, there was a question about what's your prediction about what you're going to find out with the secondary consumers, the carnivores, in terms of viability?
Yeah, I think most of them will be viable. So especially on the truffles, they have really thick cell walls, and I think that probably allows them to pass through those guts unharmed. So yeah, I'm excited to see. We have, I think, at least 15 different genera that I found in the small mammal scat. And so I'm hoping that those transfer into the carnivores, and we can do a really good assessment of which ones are viable and which ones are colonizing the trees. But I suspect that a lot of the truffles will still be viable.
Interesting. And just a quick question on why is the carnivore study taking place in the Midwest as opposed to New England?
Yeah, that's because Michael's there, and he's the carnivore guy that I know, and we met at a mammals meeting, and that's how that project got started. So
well, people were excited about the scat sniffing detecting dogs.
yeah, it's a great way to do it. The road surveys, they did a lot of driving to find those scats, and I think in basically 3 days the dogs found as many scats as they did at one site, compared to 3 sites with 3 weeks of driving. So those scat dogs are good.
Yeah, so cool. I love that. So a question came up from Janet, and she had a lot of great questions. And this one was one I wondered about because I had read that deer and white-footed mice can sometimes hybridize. So is that true? And if it is true, then how did you differentiate between them?
Yeah, that's a good question. I have seen one study from Canada, and I don't know a lot about genetics, but it might be. I guess I'm not sure on that one.
Okay.
In terms of differentiating, we used a combination of ear length and pelage or fur color and tail bicolor. So this is actually a really great example here of a deer mouse, really long ears, compared to a white-footed mice that usually has shorter ears. And deer mice have really well-furred tails that are bicolored. You can probably maybe see it here with the dark on the top and light on bottom. Whereas Leucopus, the white-footed mice, tend to be more gray on the bottom and have less hair. And so the first year, we did some genetic tests, and I think we were about 95% accurate with our IDs based on just morphology.
Yeah, fascinating. And here's a question from George, who I know is a big mushroomer. He said, let me just make sure, what about fungal parasites on truffles, like snake tongue truffle club, and how would that affect the fungal depth? Have you come across any fungal parasites?
Yeah, that's a really great question. So it used to be cordyceps. Now the name has changed and I can't pronounce it, but it used to be Elaphomyces that would parasitize Elaphomyces. And we did find it quite a bit, especially on those shallower fruiting species, but we didn't really see it on the deeper fruiting truffles. And so fruiting farther down might allow them to get away from some of those pathogens. And when those pathogens do colonize the truffles, they basically completely destroy it. So there's no viable spores usually if they're colonized early. Yeah, that's a really great
Yeah.
question.
Yes, fascinating. So this is from Joni, and I wondered about this too. So do the small mammals only consume the truffles when the truffles are in the spore phase, or do they also eat them prior to that? Like, do they eat them when they're not ripe, quite?
Yeah, that's also a really great question. So that's one of the reasons we think that those shallow fruiting truffles don't produce the same level of volatiles, because we found that even some of the maturing truffles do tend to have some odor. And once in a while, especially Macrosporus tends to be a little closer to the soil surface. And I have found partly chewed truffles that were not fully developed that were relatively close to the surface. So yeah, I think that's actually part of the reason why at least the Elaphomyces, the deeper fruiting truffles produce more odors, because if they did that shallower, they probably would be picked off before they're mature.
Gotcha. Yeah, that was great. There was a couple of questions related to that. And here's a question that I wondered about myself, and other people might too. If we were going to be out on the landscape looking around for signs and evidence of small mammals eating the truffles, would they be digging down from above to get to the truffles, or were they coming in from a tunnel and taking it from underneath? Or
Yeah, I can't answer the second part from a tunnel, but I do see a lot of truffle digs. So if you're interested in finding truffles, if you find a hemlock stand and you see digs that kind of go nowhere, usually if you put your finger in and it ends and it's kind of smooth on the bottom and you dig around that, a lot of these truffles are kind of fruit and aggregations. And so you can usually find truffles around. So I've seen
ooh,
quite a few truffle digs, and that's actually how this project got started, when we were wondering what they eat, and then we found these kind of holes going to nowhere. And at first we thought, wow, these animals are really dumb digging all these holes that go to nowhere. And then we realized that they're probably not that dumb. They're digging holes for a reason. So
I love that. That's so cool. And it gives us all something new to look for when we're out on our wanderings. So that's so cool. And here's just one last kind of question, and that has to do with birds of prey like hawks and owls. And they must also play a role possibly in the secondary consumption of the spore consumers. And are you thinking that that might be a study in the future?
yeah, that's something I would love to do. I actually had a collaborator in Wisconsin send me some Cooper hawk pellets, but it turned out that the ones he gathered, they were pretty much all eating birds and not small mammals. So I wasn't able to assess that. But yeah, I think especially owls that are eating a lot of voles and mice are probably dispersing a lot of fungi, and they have the potential to move them much farther than a lot of these carnivores even. So yeah, I would love to get my hands on some owl pellets or scat to look at that.
Oh my gosh, I just found an owl pellet today on a hike with some fourth graders. So, which just my own curiosity, if I was looking at scat or owl pellets, I wouldn't be able to see directly if the animal has been eating truffles because the spores are so small, or are there chunkier evidence, I guess.
Yeah, they usually get ground up pretty well when they're eaten, and the spores are the biggest ones are probably about the width of a human hair to much smaller than that. So yeah, you'd need a microscope to see the spores.
All right. Well, I guess I won't be doing that this summer. Wow. I just have to say, we're just about out of time, but Ryan, that was really fabulous. What an excellent job in presenting and communicating. What an exciting project too, because it has such implications in how we might choose to have best practices in forest management in the future. So really excited about the work you're doing. we all just appreciate you taking the time out of your busy schedule to share with us. And we definitely would love to have you back when you complete your carnivore study or anytime. So thank you so much. And thanks to everybody today who came and attended and have a lot more things to think about. So thanks.
Yeah. Thanks
Everyone.
for having me, and thanks for all the really great questions.
Yeah, such great questions. All right. Good night, everybody. And keep your eyes open on the small mammals.